I think with that, I will introduce our first session. So Matt Thompson from University of Alabama is doing our preliminary session here on protein structure. You know, for some of you, this is going to be review. For some of you, this is going to be stuff that you've never seen before. We're trying to sort of find a very happy medium. And so the goal of this session, or the goal of this whole program, is that, you know, you're engaged and challenged in some ways. Maybe some things are more advanced and you'll never use them again, but hopefully some parts of this will be relevant for your research and as you go on and do research in this summer. And so, but we can't talk about a lot of the tools that we want to talk about unless you understand proteins, unless you understand nucleic acids to a certain extent. And so that's why we have these first sessions. And so without further ado, I'm going to turn the stage over to Matt, and he'll tell us about protein structure.
Recording this yet?
I can cut it out whenever you want me to start.
Is he going to cut out if I come up here and say roll tide? Oh, goodness. How you guys doing? I assume you can all hear me. One of those things, huh? Is there an echo in it? I'm excited. I've said to some of the people that I know already. This is the biggest class I've got to teach since December of 2019. So, it's kind of crazy. All right, I get the wonderful opportunity to try to introduce to you a little bit about protein structure without a whole lot of background and without a whole lot of, I feel like we're doing DNA next. So this is a little bit of a challenge. I hope everything that I want to say comes across to you. So, what is a protein? This is biochemistry boot camp. Well, the answer is simple. It's the polymer amino acids connected by peptide bonds. Now you will see sort of protein nomenclature done in a number of different ways. You might see it like this top piece where you have a little bit of a line. Remember, it's a polymer, a little bit of line, and then you have like an arrow, calls it a beta sheet, and maybe another little loop and a beta sheet, and maybe an alpha helix. You might see, especially when you go in and you do the protein database exercises, you'll see these one-letter codes, right? So the protein sequence is written as one-letter codes here, M-U-Y-K. Hopefully you'll learn how to read that. Certainly will when you take a biochemistry class. So each of these little letters represents a particular amino acid, and those amino acids are strung together to make a polymer. The general structure of an amino acid is here. You can see on one side you have an amino group. Here's an amino group, so amino. On the other side, you have a carboxylic acid group. Huh? That means it cut out the first part.
Roll Tide.
He did that on purpose. Or I did that on purpose. All right, so this is where you get it, right? So no revelation. Amino acid. Amino on one side, acid on the other. Carboxylic acid. Then we have a C alpha, as we call it. And then we have, where's my organic people? You know, if I say that we have an R group, that just stands for some kind of functional group, right? So it's the R groups that change about amino acids. The rest of it stays the same, okay? And then we string these things together, we fold them up, and we make a three-dimensional structure. And here's a cartoon model of an alpha beta protein, and we'll talk about that too in a second. All right, so that's kind of the generic way. So here's the general structure of an alpha amino acid, right? I just kind of said you have an amino, whoops, let me get myself oriented up here. You have an amino group on one side and a carboxylic acid on the other, and this is your amino acid. You have the C alpha in the middle, a proton, and an R group. Now, if you're looking at this amino and you're looking at this carboxylic acid, proteins are typically hydrated, right? They're in water. What happens to this thing if I put it into water? Anybody? Let's go ahead. It forms a Zwitter ion. And why does it do that? I want to try to keep this in the general chemistry and organic chemistry fundamentals as good as I can. What might we think about when we think about what's very important for amino acids and protonations? pKa's maybe, right? Remember pKa's. And what's the pKa of an amino group? Ooh. There you go. What's the pKa of an amino group? What does pKa mean for those people that don't know? Maybe it's about 9.2-ish. I'll use ish a lot because in a protein, things folded up around each other changes pKa's. But what does pKa mean? What is the pKa? Well, I like it. We could go back and say Henderson. We could come back to the Henderson-Hasselbalch equation, and I mean he's right. pKa equals, or pH equals. I like to do it like this, so I can say pKa plus, and I'm going to use log with a base over the acid. I think over the acid, and so when something is 50% protonated, then these two values are equal, right? So this becomes 1, and what's the log of 1? 0, right? And so the pKa is actually the pH at which half of a particular group is protonated. So if the pKa is 9.2, and I'm working at physiological pH, what happens to this amino group? Remember, you might remember that there's a lone pair on here. What happens? The pKa of 9.2. At physiological pH. It's protonated. Yeah, nothing special there. It's protonated. And if this has a pKa of somewhere between 4 and 5, it's actually a little bit lower on an amino acid, but for general carboxylic acids, between 4 and 5, what happens here? Deprotonated. I like it. As long as I got somebody, I'm good to go. I need that help. I can't teach without help. See? All right, so you were absolutely right. You wind up with what is known as a Zwitter ion, or Zwitter ion. And so the amino acids are kind of dipolar when they're in solution. They have a positive side, they have a negative side. Zwitter, I think, means hybrid. And so it's hybrid ion. But ultimately, what is the overall charge of this amino acid still? 0, right? It's still neutral. Your Zwitter ions are charge balanced, right? So you have a positive side and negative side. All right. So, general structure. Now, if we were an actual biochemistry class, you guys would have to learn how to draw all these. All right? So who's had biochemistry so far? Who knows how many amino acids are there? There's 20 amino acids. Who can draw all 20 amino acids? Yeah, that's right. Okay, so look, when I teach this class, I try to give a little bit of a trick, right? It only works so far, but you can always start with C alpha and put your proton. Put your carboxylic acid and your amino group. Now, the only thing that changes is here, all right? And you can literally just go ahead and keep this same basic structure, right, as many times over as you want. Right? All we're going to do is change what the R group was. So what's the simplest thing I can put here? A hydrogen, a proton. That's the simplest thing I can put there, right? So I put a proton there, and all of a sudden I've made an amino acid. Technically, this is glycine. That's the amino acid name. There's one residue, glycine. There's a 3-letter code for it, GLY. There's also a 1-letter code for it, G. So that's easy. That's easy. Some of them make far more sense than others. Well, what's the next thing I could put here? Let's build up from a proton. What would be the next kind of thing I could put here? I can't just put 2 protons there. What's the next thing you put there? How about a methyl group? How about just a CH3? And all of a sudden, I've made another amino acid, alanine, or ALA. Maybe I just give that an A. I'll just give that an A. Well, I could do it again. Just keep building them up. Now, if anybody actually knows all 20 amino acids, you know this whole build-up process fails eventually. It seems cool while I'm doing it at first. It's an easy way to start remembering them and writing them down, but it doesn't always work. All right, so what can I do then? Well, I could say, well, CH, and then I can put 2 methyl groups on there. And all of a sudden, I've created valine, or VAL, or V. So that's easy. And this can go on and on. You can get it a CH, whoop, that should be a 2. How about a CH2? And then we'll put this ring on here. We'll put this ring on here. Now check this out. Now what's the name of that ring? Huh? Benzene. There's another word we use to describe it. What else? What else do you call it? Phenyl, right? Right. You call it a phenyl. So here is a phenyl. Look at that. Alanine, because that was the alanine, just the carbon, right? So there's your phenylalanine. We call that PHE. And I think we just give that an F. All right? And so you just build these amino acids up, right? There's a general structure. Start with C alpha, and then add in your proton, your carboxylic acid, and your amino group. And all you have to do is know the side chains. Pretty easy, huh? All right. Here's 9 of our amino acids, right? So amino acids are kind of separated into non-polar amino acids, polar uncharged amino acids, and then polar charged amino acids. Now I know this is a little bit tough for some of you to see. It should be in your slides. But here are the 9 non-polar, I'll just call them AAs at this point. How about that? 9 non-polar amino acids. And if you look at these, they pretty much have what we call aliphatic side chains, right? Or we have some aromatic side chains. All of these things, if I said they were non-polar and I was giving you aliphatic side chains or aromatic side chains, would you say those were hydrophobic or hydrophilic? I would say they're hydrophobic. Yeah? All right. So these are the hydrophobic residues. All right? Now, if you really want to do this properly, then you would go through, learn all the structures, how to draw them, learn all the names, learn all the 3-letter codes, and learn all the 1-letter codes. I try to say learn instead of memorize, even though for the most part you're memorizing. We don't like to use that word. You got to learn. And we won't do it right now, but you'll see why. How many of you have read journal articles where it uses the 3-letter code? Yeah. How many of you read journal articles where it uses the 1-letter code? Right? How many of you have read journal articles where they don't keep it consistent? It kind of goes back and forth, right? So you sort of got to know all of them. It's just one of those things. It's learning the language of proteins. And if you take my class next semester, you'll learn all these things. All right. So those are the non-polar ones. Then we have uncharged polar side chains. Uncharged polar side chains. Now again, it's a little bit smaller than I anticipated, but here we can see one, and I'm just going to draw the side chain on it. This is a CH2OH. So it's serine. It has an alcohol on it, right? And of course, an alcohol, you might say, well, the OH is like this, so oops. The OH is like this, so it has a dipole moment. It's going to be protonated at physiological pH, but it has a dipole moment, so it is a polar uncharged amino acid. There are 6 of these. We say polar uncharged. And then we have 5 polar charged. Now, coming back to that whole pKa thing, I want to show you, you notice this column right here, this column is pKa of the side chain. And you'll notice this column is empty here, right? Because these are the non-polar amino acids. And then when we come over, and it's a little bit challenging to see, but you start having side chains that can be protonated. They can donate a proton, or they can take a proton from maybe a substrate, right? So these are the ones that have exchangeable protons or hydrogens, right? So you can take a proton off, or you can add a proton, then it becomes charged, right? Does that make sense? Are we good enough for right now? I just want to, this is going to be important later when I come back. I know it's hard. There's 20 amino acids. I spent a whole lecture on all 20 amino acids when I actually do this class, and now I just gave it to you in 5 minutes. So take it for what it's worth. All right, but if I can get anything across to you right now, it's really to think about ones that are hydrophobic. What would you call these then? Yeah, sure. So here we have hydrophilic. So we want to think about, for the sake of today, we want to think about hydrophobic, we want to think about hydrophilic, and we want to think about being able to donate or abstract a proton. And I think those are very important aspects to carry with you right now. All right. So just some basic amino acid nomenclature. Now this is hard. This is hard, so bear with me, all right? I already told you that the general structure had the amino group and then a carboxylic acid. Now this is meant to already be a peptide bond, and I'm going to get to that in a second. And I said there was a C alpha. Well, if that first carbon is the C alpha and it has an amino group and it has a carboxylic acid group, what's the next carbon? If I start adding onto this chain, what would you call the next carbon? C beta. Then what? Delta, gamma, epsilon. I mean, stay with me. All right, so just some general nomenclature when we start talking about things. It will be important for some of you. Maybe as you start talking about your projects, you know, you guys don't have to have all this stuff memorized, but at least have a general understanding of it. Just know where it comes from. Some amino acids, well, in particular, one amino acid, cysteine, has the ability to form a disulfide bond, right? Cysteine can form a disulfide bond. This becomes important for structure, and I will mention that again. So how does a cysteine form a disulfide bond? Well, you can if I oxidize something, what have I done to it? Oxidation is loss of an electron or something like that, so lose an electron. All right, so in the presence of oxygen, if you look at something, and I'm going to do this from like a super simple gen chem, if I just drew maybe something like a Lewis structure of oxygen, you guys remember those? Or maybe I just put a couple of lone pairs. How many electrons does it have? 6. We can draw it like that. And so you can imagine that, you know, if it took an electron here, and maybe it took an electron with a proton, I'm going to kind of redraw it like that. So now I have an S dot maybe. And then this comes, and then I have, you know, one there, and then maybe I take another one here. I take another proton and an electron, and I have an S dot. Redraw it. Ooh, that's a dot. Sorry. So now I can make a water, but then what can happen here? Form a new bond, right? This is a disulfide bond. Oxidation of cysteine, 2 cysteines, and you can get a disulfide bond. All right. Do you suppose that if I'm discussing things like protein structure, do you suppose that disulfide bonds matter? Like, do you suppose that this makes things more rigid? Or, no, maybe I shouldn't say rigid. Do you suppose this would make things more stable if you had a folded up protein and then maybe you had a stitch where you kept it together, right? So that's important. All right. Now, I used this because I really like these tables that have all the pKa values, and they keep everything drawn in the right charge states, the Zwitter ion. But, you know, for the sake of people who've at least had gen chem and you've seen space-filling models, right, they're not just sticks drawn on a page, right? There's electron density around all these nuclei. And so when you look at them, right, they're going to take up 3-dimensional space, right? Here's the acid side, the carboxylic acid side. Here's the amino side. Here was C alpha, and then there's a methyl group that was alanine. Here it is space-filled, right? Remember, this is non-polar, so this was hydrophobic, right? This was hydrophobic. And then here's another one, isoleucine, again, the carboxylic acid. And these things, they have volume. I teach gen chem, so I also know that we teach space-filling models. Everyone's at least seen space-filling models, right? And so just some examples. Here was serine. I already talked about it. The carboxylic acid, the amino, C alpha, and then we said there was a COH, and so there's a dipole here with maybe a partial negative charge and a partial positive charge. And then Glutamine has an amino here, and then a carbonyl. This is actually a double bond. So these aren't protonated or deprotonated, so these are polar and uncharged. And we said these were hydrophilic. And then, of course, charged polar side chains. But they all have space. And when you string these things together and you fold them all up, they're going to have a larger volume that is the protein structure. And you can see here, red is negative and blue is positive. The white is somewhat neutral. But remember proteins are hydrated. These things are in waters. And so if I just sort of draw my generic water molecule like this, and I might say there's a dipole moment that way, I can just draw them as a V, where this is a negative side and this is a positive side. What's going to happen? This thing is going to get hydrated by water molecules that neutralize the overall charge of it, right? And you're going to see these polar uncharged or polar charged residues surrounding the outside so that it can interact with water, all right? Where are the hydrophobic residues going to be, you think? In the middle, and I'll show you that too. All right, questions, comments so far? Everybody with me? Oh, yeah.
When you call a carbon like an alpha carbon, is that just like another way of like numbering them, like you do in organic with the carbon backbone?
Yes.
Okay.
We use alpha, beta, gamma.
Okay.
Alpha, beta, delta, gamma. Epsilon. It's in there. Yes. And using, Chris will talk about the numbering of DNA bases. And so, yes, we use Greek letters for amino acids. We use numbers and prime numbers for DNA bases, and then for sugars, respectively. So it helps you keep it a little bit track instead of just calling everything carbon 1 and carbon 2. All right. Let's see if this works. So how do we arrive at this three-dimensional protein structure? Where does this come from? And this is where it really comes down to chicken or the egg kind of thing, and what do we do? And which made this lecture a little bit challenging. So I want to show you, if I can, briefly, and I won't run through this whole thing, but I always just find this fascinating. Proteins are made in the cells by something called the ribosome. Now, we haven't talked about mRNA or tRNA. mRNA is messenger RNA, tRNA is transfer RNA. It'll talk a little bit about that. If a picture is worth 1,000 words, these new videos are great.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome.
So for those that don't know, mRNA is what is actually transcribed from your genome. mRNA, it's messenger RNA. It carries the code that's going to get read to make a protein.
Production of a particular protein that this gene codes for. There can be several million ribosomes in the typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic.
All right, so that was supposed to be the protein being made coming out the ribosome, the red.
To assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building.
All right, so for the sake of just reading this, or looking at this, the little red piece is the amino acid, right? So in all that, the little red piece is just the amino acid.
That's the small glowing red molecules are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positioned to carry the gene
Oh no,
message,
sorry.
known as a codon. Each codon unit of the ribosome.
I just want to stop it right here. because it's basically cutting the ribosome in half and showing you what's happening. So these tRNAs come in one at a time, and they have an amino acid on the end, and this ribosome, you got to think about, there's another half on this, right? We got to slice through, okay? Each 3 nucleotides that come through here represent a codon that match up with one of these tRNAs, and then there's amino acid on the end of it, and those amino acids get stitched together in this protein. Well, now, if this protein's making proteins, where did this protein come from? It's just a thought. So it's a fascinating process, right? You have a protein building proteins, one amino acid at a time.
So that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA gets ratcheted through the ribosome, the mRNA sequence is
All right, so here's our little string of amino acids coming out. That's all I'm going to use for this video. Is everybody with me? Still, though, think about the question. If it's a protein making a protein, what made the protein? All right, so that red string of amino acids coming out of the ribosome, right? That is your polypeptide chain, or your amino acid chain. Each one of these little balls represents an amino acid. That is a generic way. Here we had PHE. What was PHE? That was phenylalanine. It was also an F. So if I wanted to, I could draw a side chain there, right? It's not just the little ball, it's a side chain. Make sense? All right, great. So, how do we form these bonds between these amino acids? Well, if you go into any biochemistry textbook, it will tell you that there is a condensation reaction, right? This is we have 2 amino acids. Here's the amino group, here's the carboxylic acid group, here's the side chain. Here's an amino group, here's the carboxylic acid group, here's the side chain. These 2 things come together to form a new carbon-nitrogen bond, and out comes a water. That's a condensation reaction. It's also known as a dehydration synthesis. Now, who's had organic chemistry? Excellent. Can somebody look at this and propose any kind of logical reaction mechanism from this figure that's in every biochemistry textbook? Where does that water come from? Well, Thompson, it's highlighted in red. It must be that oxygen and that hydrogen and that hydrogen. And out comes the water. Okay. I'm going to form a bond, right? If you're an organic chemist and you're going to form a bond, you're probably going to have some kind of attack, aren't you? What kind of attack? You might have some kind of nucleophilic. Well, somebody tell me where the nucleophile is here. Huh? You guys like the oxygen? All right. Oxygen is now my nucleophile. What's it going to attack? Huh? So if my oxygen attacks a proton, then what do I make? I make an oxygen-hydrogen bond. I need a nitrogen-carbon bond. Yeah. Doesn't make a whole lot of sense, does it? All right? Okay. So let's think about it a little bit different. What if, all right, what if we say, and I use my notes some on this because I want to make sure I keep it all right. I'm going to say, Now this might take me a second to get all this drawn out. I'm going to call that R2. And I may not have time to do all this. All right. What did we say? I harped on it when I was talking about polar charged amino acids. They had what? Exchangeable protons, right? They had exchangeable protons. And we just learned that this whole formation of a bond happens inside the active site of a protein. Now, typically, inside the active site of a protein, you have amino acids that can have exchangeable protons. You know why? Because exchangeable protons can do chemistry. And so, if you're looking at this, I probably need to draw my minus here, don't I, to keep this right? Right? There's maybe when this amino acid enters the ribosome, something gets protonated and something gets deprotonated. Right? We have this Zwitter ion. What could we protonate? Could an amino acid side chain maybe protonate this water? And then maybe deprotonate? Oops. Maybe deprotonate this nitrogen. Then what happens to this nitrogen? All of a sudden, if you calculate the formal charge on it, right, you're going to have a lone pair there. Formal charge now would be 0 if you did it. But now, do you have a nucleophile? Do you have a nucleophile?
Yes.
Which one is it?
Nitrogen.
Yeah. What's nitrogen need? I mean, what's the nucleophile need to do this attack? Organic chemists. Electrophile, if you will. Is there a good electrophile in here somewhere now? Which one is it? The carbonyl carbon. And so now you can have nucleophilic attack of the carbonyl carbon, right? And I won't go through this whole mechanism in the interest of time, but you can imagine, well, that's supposed to be an arrow, not a bond, right? You can imagine that, you know, when you do a nucleophilic attack on a carbonyl carbon, right? Maybe you guys drew this where your electrons from the double bond go up, and now you have now you have 4 bonds to this carbon, and you have your electrons out like this, right? And then, I'll go ahead and, is everybody okay if I really, really shortcut this and just say, well, you know, now I have my nitrogen here, right, plus R. It would be all of this other stuff, if you will, right? And so maybe if I was really trying to keep this, maybe I'd have them both still here. Maybe my electrons could actually return. If you think about this from a really organic, you know, fundamental kind of thing, then I might get that. And then I'd reform this double bond, and maybe these electrons would go to the oxygen here, right? And then this would leave. So OH. And OH is a leaving group, maybe. Is OH a good leaving group? No, it's not. Right? It's not. But you can imagine that you'd have this OH, and, you know, maybe it could extract a proton from the nitrogen. Maybe it takes this proton, or I'll make sure I draw it the right way. Maybe it takes this proton, and then these electrons go back to the nitrogen, and so ultimately what you would wind up with is the water molecule going away, and your nitrogen with its 2 electrons, and your new peptide bond. But the hydroxide ion is not a good leaving group, you said, so what gives? I'm sorry?
We protonated the alcohol group again, and then there was an oxide ion, and that's us.
Yeah, I would just argue that it was happening inside the active site of an enzyme, and that's what they're made for. And so they can stabilize poor leaving groups by developing sort of they can develop polar bonds and even some degree of charge separation to encourage leaving groups, right? Right now, we do not know exactly what happens at the inside of the ribosome, but this figure is misleading. All right? It's not a true representation of a condensation reaction or dehydration synthesis. All right? So this really what happened, and we form a new peptide bond. That's how peptide bonds are made. Okay, so then we have a peptide bond, or we have one mechanism potential of a peptide bond. Right? If you were an organic chemist and you drew this out on paper, you would be criticized because someone would say, no, that's not a good leaving group. It doesn't work that way. But as a biochemist, you can go, nope, all bets are off because we're inside the active site. Things are different. Proteins can even modulate pKa's, right? So what you would think about in a pure solution can be completely different inside a protein. Right? All right. So peptide bonds. Peptide bond, it's a single bond. Single bond. We're free to rotate here, right? It's a single bond, you can just rotate around it. No big deal, right? Nope. Turns out it's more interesting than that. All right, so if you look at this, if you look at a peptide bond, and I want to, like here's a carbon alpha, here's the peptide bond, and here's a carbon alpha, okay? And if I just sort of drew it over here off to the side, and I'll shorthand it a little bit just to save time. I'll put my proton down here. That's a nitrogen. And then C alpha and an R group. All right. So there's a couple of electrons here, right? What do you guys, if you were looking at this, and you had organic chemistry class, and you were looking at this from a delocalization kind of thing, do you see something that can happen here? Is there any kind of resonance that can take place? Right? So you can actually have some electron donation into this bond, and then these electrons can kind of move up to the oxygen, where you wind up with, and again, I apologize for my sort of quick trying to draw this out, but you might have something that actually looks like this, where there is double bond character at this peptide, or peptide bond. And so once it has some double bond character, then can it rotate? No, and it turns out that it's about 40%, double bond character, roughly, right? So if it's 40% double bond character, then it actually turns out that it's rigid, and it turns out that they're always trans. It's always the trans configuration. Trans configuration with respect to the peptide bond, and what are we talking about? Right? This C alpha is trans to this C alpha, as opposed to what? Cis, if they were cis, right? So if we're looking at, if my thumbs are the peptide bond, then my fingers are the C alpha, or where the next residue is. They're always opposite of each other, right? They're trans. They're not cis. Okay? How do we know that? Well, from measurements, about 1951, Linus Pauling and Robert Corey, I think, maybe, I don't know. I do my best when I teach the class to remember some of these things. But it turns out that this the carbon nitrogen, if it was going to have double bond character, and you were looking at bond length, if you added in double bond character, what happens to the bond length? It's going to get shorter. And it turns out that the carbon-nitrogen bond is about 0.13 angstroms shorter than what is expected. And then my carbon-oxygen bond here, or actually I should say my carbon-oxygen double bond, what's going to happen to it? It's going to get a little bit longer, isn't it? So it's about 0.02 angstroms longer. So in fact, you can see this, right? There's double bond character in the peptide bond, and what happens then if you look at the extended conformation of a polypeptide, or what we call the protein backbone, this is the protein backbone all the way down through here, right? Your residues wind up on alternating opposite sides, all right? And then just for some nomenclature, your first amino acid in any polypeptide chain, the first amino acid in any polypeptide chain is going to have this amino, and the last amino acid in any polypeptide chain is going to have this carboxyl group. And so we say that any polypeptide chain, and maybe you guys have heard this, it has an N terminus. I'm just going to say N term, and it has a C terminus, right? So any polypeptide chain has an N terminus or a C terminus just by the nature of how the peptide bonds form, and they're always in the order. And then you have planarity about each one of these peptide bonds, and that gives you this alternating, repeating, if you will, pattern of where your side chains are. Now, if you're looking at this, and I have a planar, this is going to fail. If I have a planar piece like this, and I have another planar piece like this, and I can rotate here, what happens when I get over on this side? Right? If I have these two, like think of the two planar green pieces, right? I'm like this. I can rotate like this, but ultimately they're going to, whichever way I went, they're going to run into each other when they get over here, right? So it actually, if you look at where this little rotation point might be, you can't go all the way around it because of steric interference, right? So this planarity sets up rigidity, and you can't even rotate all the way around. So we refer to this, here's the alpha carbon, the C alpha C, which would be this one. We refer to this rotation angle as psi, and the C alpha to nitrogen, which is the actual, well, this isn't. Here is a peptide bond, and here is a peptide bond. These are double bonds that generates this sort of planarity of this segment, and here is the rotation point. But you can imagine that you can't go all the way around, or these planes are going to run into each other. And so this C nitrogen is phi. So we talk about psis and phis, right? These are the rotation angles of the backbone. I already told you, you can't go rotate all the way around because of steric interference. They run into each other. So what does that mean, ultimately? Can the backbone adopt any configuration it wants to? No, right? That's really what it comes down to. You don't just get any configuration you want to. It turns out that with respect to allowable phis and psis, rotation angles of the protein backbone, there are really only 3 small groups of angles that work. So this is a plot of here is phi, and here is, here's psi, here's phi. But you'll notice it goes to plus 180 and minus 180 for phi, and it goes to plus 180 and minus 180 for psi. And that's basically referring to the fact that starting from planar, that's 0. And so you can rotate one to 180 degrees, or you can rotate one to minus 180 degrees, and you can rotate the other one to 180 degrees, and you can rotate it to minus 180 degrees, okay? So if you plot where you can fit these things, there's really only 3 relatively small areas that work. We won't worry about this one so much, but we'll worry about these two. And it turns out that these 2 areas, all this other space in here, this, I don't know, light gray, grayish brown, this is all forbidden. Forbidden in that it just can't adopt that geometry, all right? And so there are really only 3 types of geometries that the proteins can adopt. One of them is a beta sheet, and the other is an alpha helix. So let's talk about some simple hierarchy of proteins then. Protein structure. All right, there are 4 levels of protein structure. I'm going to go through each one. 4 levels of protein structure. Did you have a question? No. The first level is what we call primary structure. Primary structure. Sometimes we represent that as just one knot, right? Primary. Second is secondary structure. The secondary structure, right? So you can imagine primary structure is just the sequence, right? Secondary structure comes from this plot. This is called a Ramachandran plot. But the secondary structure comes from what is allowable for the protein backbone to actually adopt. So there are only certain, we're only going to talk about two secondary structures, the beta sheet and the alpha helix. So we have primary structure. Secondary structure is what's allowable. And then you can imagine that these pieces of secondary structure can fold up on top of each other to form the three-dimensional structure, which we call tertiary structure. And then there's one more level, and that is for multi-domain proteins, where I have one separate protein and another separate protein, and these two can come together and form a multi-domain larger protein, and we call that the quaternary structure. So we'll talk about each one of these. How are we doing on time? Perfect. Perfect. Primary structure is the sequence. Primary structure is the sequence. This always baffled me. I was like, why do we call that structure? Right? Why do we call a sequence structure? Right? Why does it matter? How is a sequence structure? How does a sequence mean something? It's just a chain of amino acids. Why does a sequence mean something? That's just a string of letters, isn't it? Nothing special about it whatsoever, is it? Just a string of letters. It's the same string of the same letters, right? Do they mean something different? Yeah. Order matters, doesn't it? Order matters a lot, especially when you start thinking about how a protein might fold. If I wanted a protein to start folding and I was making it in water, and I wanted it to start folding, what kind of amino acids would I put out there to get it to fold, start folding? Huh? Hydrophobic, right? You want those to start folding up, right? So sequence matters. It's primary structure. Another thing that's included in primary structure is those disulfide bonds that I talked about. We sort of include disulfide bonds in primary structure. Might see it drawn out like this. Now, obviously, this disulfide bond is not that long, but what's that going to tell you about those two cysteines? When this thing folds up, I bet they're right next to each other, don't you think? Otherwise, there's not a disulfide bond. All right? So that's primary structure. The sequence is primary structure. Then we move to secondary structure. How many allowable secondary structures did I tell you there were? I said there was 3 small areas, right? One of those small areas is really a lot smaller than the other one. It's a left-handed alpha helix. We're going to talk about right-handed alpha helix. So if I just said, don't distinguish between left and right alpha helix for right now, how many types of secondary structure are there? 2, and they are? All right, people know. All right. So secondary structure, then it folds over to tertiary structure, and then we have quaternary structure after this. Let's talk about each one. The alpha helix. The alpha helix is pretty amazing. If I asked you, what's the importance of a hydrogen bond? If you came out of general chemistry, what's the weakest bond? Well, those pesky little things must be pointless, right? They don't do anything. I want a strong bond. What do I got? If I want a strong bond, what do I get? What do I need? Huh? I mean, I would think like a triple bond or something like that, right? That's a strong bond. Hydrogen bond, those are pointless. Not really, right? Not really. So look at what a protein can do, all right? So this is the base, this is an allowable structure of a protein backbone. It's an alpha helix. We say that this is right-handed. Right-handed alpha helix. That means if you're kind of looking at it like this, it curves up. It's like it's your right hand. If you point your thumb up, it's your right hand. It's right-handed alpha helix. It has optimal phi's and psi's. The phi is about minus 57 degrees, and the psi is about minus 47 degrees. For each turn of this alpha helix, there are 3.6 residues per turn. If I look at a turn, I'm going to say maybe from here to here, that's one full turn. That's known as the pitch. And the pitch is about 5.4 angstroms. Okay. That seems like a lot of pedantic stuff. I make my biochemistry students learn it. But here's what's cool. If I look at this, now it always fascinates me that it turns out that this is what's allowable. Like, I mean, I would argue it probably evolved this way, but that's just me. If I look at this alpha helix is held together by this. This is a carbonyl from the backbone, and this is a nitrogen and a hydrogen. And so now I have a hydrogen on a nitrogen, and I have a carbonyl oxygen. What can I form between them? What can I form between this oxygen and this hydrogen? A hydrogen bond, right? So this wimpy, pesky little hydrogen bond is actually one of the things that's holding my protein together. Is one hydrogen bond a lot? Not really. But look what the protein can do. Every single carbonyl is hydrogen bonded to an NH of another residue. And it turns out that it is, we say it like this. It's the CO of the nth residue, and that is, I'm just going to say H bonded to the nth, maybe I'll say nth plus 4 residue, the nth plus 4 NH. But it's not just, right? So basically, if I was to count, if this was carbonyl 1, I'll just say it's carbonyl 1, then there would be, let me erase some of this so I can see it. I don't want to lie to you. If that's carbonyl 1, there's the second one, there's the third one, well, I guess I should say it like this. It's this purple residue, 2, 3, 4, and 5, okay? So that you get to the fifth one of the residue. But it's not just one every 4, it's every single residue. In the alpha helix, with the allowable phi's and psi's and the way that it folds up, it's not just that you get one hydrogen bond every 5 residues, you get a hydrogen bond at every single one. It maximizes all the hydrogen bonding capacity in the backbone. Now, if I fold up a protein and I have hundreds or thousands of hydrogen bonds, all of a sudden I've got something that's really strong, right? That pesky, wimpy little hydrogen bond becomes one of the most important things for biological molecules. Not just because of by itself, but the sheer magnitude, or the sheer number of them, if you will, within a protein, okay? If I look at this, so the purple balls are supposed to represent my side chains, and I kind of want to go, I'm going to erase it like this, get rid of some of this. So if I draw where these purple balls are, so this is supposed to represent a side chain, and this is a side chain, and then this is going back in the page, that's a side chain, and that's a side chain, and that's a side chain, and there, and there, and there, and there, and there. What can you guys tell me about these? Where are they with respect to the alpha helix? Are they on the inside or outside? They're on the outside. And then look, they're all kind of pointed in the same direction, if you will, right? All right, that's kind of neat. And now when you look at a space-filling model of it, the yellow is actually, see here it was just supposed to represent an R group, if you will. But here you actually have a space-filling model, and you can see that all the side chains are kind of pointed to the outside, and all the backbone is on the inside. Right? So even when you make an alpha helix, you've basically capitalized on the ability to put all your side chains on the outside. Right? Everything that might have a functional group, everything that might do chemistry, everything that might, interact hydrophobically with other residues. They're all on the outside, and I'll show you that in a second. So another secondary structure is the beta sheet. Now, beta sheets, beta sheets don't have that N to N+4 type of hydrogen bonding pattern. But when you have a beta sheet, right, so there are two kinds of beta sheets. We can have anti-parallel beta sheets where, the two, well, it's just that. The two sheets, one is moving one direction from N terminus to C terminus. Remember, we labeled N terminus to C terminus, and then the other one is going in the other direction from N terminus to C terminus. These are anti-parallel beta sheets. At the same time, we can have parallel beta sheets where this is the N terminus and this is the C terminus, and then also this is the N terminus and this is the C terminus. Make sense? Anti-parallel or parallel. Beta sheets don't have that same sort of N hydrogen bonded to N+4 residue, but they still maximize the hydrogen bonding capacity between the two chains, right? There's not a quote-unquote open hydrogen bond in these secondary structures, whether it's alpha helix or a beta sheet. That's amazing. That's absolutely amazing. And so in order to maximize that hydrogen bond capacity, it's not just this flat little piece. It actually kind of pleats a little bit. So it pleats some to maximize all the hydrogen bonding interaction between the two sheets. And now the purple is supposed to represent my R groups again. And if I just look at one sheet down this side, here's a purple on this side. And then the next residue, oh, it's on the other side. And then at the next residue, oh, it's on the other side. And so it turns out that on a beta sheet, the side chains are perpendicular to the beta sheet itself, sticking out on either side. You have to admit that's kind of cool, right? The two allowable secondary structures fold up so that you have an alpha helix where all the side chains are exposed and it maximizes hydrogen bonding interaction, and then the beta sheets all come together and they pleat up so that you maximize hydrogen bonding interactions and all the side chains are exposed on either side. Wow, we've made some fantastic building blocks, haven't we, for a protein? All right, so how does that look? Well, you don't get the whole space filling model, but if you think about this, if this beta sheet is in the plane of my slide, then basically you're seeing all the purple residues on this side. And they alternate, right? So you have one on one side, then one on the other, then one on one side, and one on the other, and there's 7 angstroms here between the two pointing out on the same side. Everybody got that? Okay. All right. All right. So beta sheets are connected by beta turns. These come in a number of different ways, but just for simplicity, we have something called, well, beta turns reverse the direction. And when they reverse the direction, so this, I guess it doesn't matter, but you can imagine let's take this beta sheet running this way, and then imagine that right here is where we start our turn. What is stabilizing our turn? Hydrogen bonds, right? So even in this reverse turn, there's a hydrogen bond. This is for an anti-parallel, where you just have a reverse turn. Sometimes you have, this would be a parallel beta sheet, Now, what might stabilize these two parallel, what right here might stabilize these two parallel beta sheets? What if I actually put an alpha helix in there that laid down across the top of them? Right? And this is how protein structures are generated from just the two secondary structures, alpha helices and beta sheets. Okay, so here we go. It comes down to things like this simple. If you're looking at this, the main chain is gray. That's the gray stuff. The backbone, that's gray. Nonpolar molecules are gold. All right, well, it doesn't matter. You gotta pick it either way. Nonpolar molecules are gold. And let's say polar side chains are purple. All right, everybody with me? So I got an alpha helix. Here's an alpha helix. Here's a beta sheet. Let's suppose I could just take these two pieces of secondary structure and throw them in water together. What would happen? If I could maintain their structure, unfortunately, it wouldn't be so simple as just putting them. I mean, just bear with me for the sake of the exercise. What if I could throw these in water together? What would happen?
The non-polar side chains fold inward.
Or maybe the nonpolar side chains from this alpha helix start to interact with nonpolar side chains on this beta sheet, and then we have sort of an alpha helix forming next to a beta sheet, right? I mean, that's it. It comes down to things being that simple. You have your secondary structures that maximize the hydrogen bonding interactions, present all these side chains in such a way that when they start to be in solution, you can now start to have this beta sheet interact with this alpha helix in such a way that the nonpolar molecules will interact with each other, and the polar and charged molecules will be on the surface where the water is. And sure enough, that's exactly what happens in proteins. If you take a whole protein structure, this is just a cytochrome protein, and you look at this, the hydrophilic molecules are green, and if you can see it, that's all these. And then the hydrophobic molecules, or hydrophobic side chains, I guess I should say side chains. Hydrophobic side chains are orange. And where are all they? They're all on the inside. I would love to just be like, it's that simple. But, I mean, arguably it kind of is. You know, maybe there's more to it. I think from an introductory standpoint, it's kind of that simple, all right? We take these secondary structures and we form from the secondary structures what are called supersecondary structures or motifs, right? And so these motifs, this is relatively simple. This obviously would be something like a beta, alpha, beta motif, right? And if it was just this motif, you might say that if you're looking at the plane of the slide here, on the top of this arrow, this represents the beta sheet, maybe there's hydrophobic residues, and on the top of this arrow, maybe there's hydrophobic residues, and then on the bottom of this alpha helix, maybe there's hydrophobic residues, and that's how these secondary structures come together, right? And then, you know, this is just this is a beta hairpin type structure where it has these reverse turns in it that we just talked about. I remember when I used to write a journal article about my protein, I would say it has a tandem beta, alpha, beta, beta, beta motif. I would say that my protein had a tandem beta, alpha, beta, beta, beta motif. That was its structure. And now you guys would know, well, that's a beta sheet connected to an alpha helix. Probably looks, if you drew it the way I introduced it at the beginning, it probably looks something like this, with 3 arrows, right? There's my beta, alpha, beta, beta, beta motif. And then I said it was tandem, so what does that mean? There would be another one. There's 2 of them hooked together, right? So this might seem oversimplified. This is beta, alpha, beta. This is a beta hairpin. What am I going to call this one? Anybody? Well, we might just call it alpha helical. Helix turn helix, or alpha alpha, whatever you want to do, yeah. One of them that I've never been able to draw is this thing called a Greek key. This is supposed to be a beta sheet with a beta sheet, and then a hairpin, and a beta sheet, and a beta sheet. And then right here, you're supposed to fold it back over on itself so it forms this sort of reverse turn, all the way back over to the other beta sheet. It's called a Greek key. Greek key. Now, these things, these super secondary structures, they sort of repeat in proteins, right? They show up. They're kind of universal, if you will, right? And then this is protein classification. Well, they're either alpha helical protein, or they're beta sheet proteins, or they're alpha beta proteins. There's not a whole lot to it, right? Those are only 2 secondary structures that we're using. So, sort of some unique, these are some unique things are sort of 8-stranded beta barrels. Have you guys seen beta barrel proteins? I think these are so cool, the way they sort of roll up. Just 8-stranded beta sheets. Sometimes here's just that reverse turn, reverse turn, reverse turn, reverse turn. Here actually is one that has a Greek key. We might say this is an alpha beta barrel because it has alpha helices in between each one of its beta sheets. All right, so these secondary structures are what fold up into the 3-dimensional protein structures. I just like beta barrels. This is always one of my favorite proteins. This is green fluorescent protein, and you can stick this onto other proteins, and you can track where other proteins go because it glows. It's just neat. This is a beta barrel. A protein looks like this, right? A protein looks like this, and it fluoresces green. It's really cool. It's really cool. We can take those simple super secondary structures or those motifs and put them together, and all of a sudden we have 2 domains. Right? So now this is a 2-domain protein. It's really just one super secondary structure connected to another super secondary structure, if you will. If anything comes across, this is amazing. But do you see the simplicity with which this happens? You string some amino acids together, they have only certain conformations they can adopt, and then they fold over on themselves, if you will, after they make their secondary structures, and then those pieces can be stitched together in different ways to form different proteins, right? And then as things evolve, and mutations in your genome happen, so if a mutation happens in your genome, for those of you that are biochemists, if a mutation happens in your genome, then that actually is going to change what your messenger RNA is, because your messenger RNA is going to read the mutation. Off comes your messenger RNA with an incorrect codon now. And so if your messenger RNA has an incorrect codon, what ultimately changes? The protein, right? The protein. And so across organisms and over time, the sequence might change, but if we just look at things from eukaryotes to prokaryotes, structure is conserved. So even if you undergo a mutation across organisms and you have a little bit different protein sequence, sometimes even a lot of bit protein sequence, right? A lot of times amino acids in the middle are always conserved, and if you look at this across organisms, this is a particular protein across organisms, from eukaryotes to prokaryotes, structure is conserved, right? It is the structure of a protein that is conserved far more than a sequence for a particular function that it has to do. All right. So that's how you can actually, there's no way to avoid mutations in your genome over time or throughout evolution, right? But as long as structure is maintained for a particular function, that function can continue even if the sequence is changed. The last little bit of structure that I want to talk about is quaternary structure, and I'm not going to give you any more than this on that, because we've talked about primary structure, that is the sequence. We talked about secondary structure, those are the alpha helices and those are the beta sheets. We talked about tertiary structure, where some of those super secondary structures can fold up into maybe domains, and domains can get stitched together, and then if we make a protein and we take multiple units of the same protein, or even units of a different protein, they don't have to be the same, and we add them together, this is actually the blue, the green, and the yellow, and I guess the lighter blue. These represent four subunits, four different proteins. I also shouldn't say 4 different proteins, they're 2 different technically. But the 4 subunits come together to form a larger protein, and this is quaternary structure. And that is the hierarchy of protein structure. Where's my inorganic? I always like this one. Where's my inorganic chemist? Anybody had inorganic? That's why I love biochemistry. You can talk about Henderson-Hasselbalch equation, and you can talk about organic chemistry, and then you can put proteins together, and you could say, hey, if I look right down in the middle here, and I draw a line, I got a rotation, and that's a C2 axis. For you, who loved all the symmetry and things? Oh, come on. Yeah, see, you can start putting these things together in these oligomeric proteins, and you can even think back to even some of your inorganic chemistry and symmetries and all that kind of stuff. So that's one of the reasons I love biochemistry. You can put a little bit of everything in it. And so with that, proteins, we basically said, are just chemical chains made up of 20 amino acids. 20 amino acids, string them together. They can form, well, the backbone, the planarity of the backbone, right, and the trans configuration of a peptide bond, that double bond characteristic, has consequences, right? Helices, alpha helices, beta sheets, and then, of course, those turns that we mentioned in there. Protein structure, simple. Primary structure is the sequence. Secondary structure comes from the consequences of the backbone. Tertiary structure are some of those super secondary structures folded up. And then quaternary structures when different proteins come together. So welcome to this. I hope that helped to a degree for an introduction to protein structure. That was a lot in one 75-minute talk. So thanks for being here. Hope you guys enjoyed everything. Roll Tide.